It's Dr. Centeno, and I'd like to talk today about autologous versus allogeneic mesenchymal stem cells. Now, there's a lot of misinformation out there about this topic, and I think some of that got going by some of the lectures that we all heard through the years at regenerative medicine conferences. And the story went something like this, right? We shouldn't call them mesenchymal stem cells, we should call them medicinal signaling cells. And that data was based on some studies that seemed to show that the mesenchymal stem cells that were being used in animal studies didn't stick around, but that they released paracrine signals to help with repairing tissue. Now, the problem with all of that is that at the first level, that made sense. But when you peeled back the onion to understand it at a deeper level, you saw a problem with that statement. And the problem with that statement is simply that if you look at animal studies, and most of them are done in small animals, rats, rabbits, etc., because the animals are so small, it's impossible for researchers to get enough bone marrow from one animal and use it in an autologous fashion. So almost all of those studies are done with allogeneic mesenchymal stem cells derived from bone marrow. And that's because you have to pool bone marrow of many different rats, for example, in order to get enough to treat one rat. So you basically have cells that are foreign to the animal, and the animal's killer T system takes those cells out. Now, why is that important? Because in these references right here that you see on the screen right now, we have a completely different picture for autologous mesenchymal stem cells. So if it's the patient's own cells, then these tracer studies show very clearly that unlike the allogeneic cell data that we heard all about at conferences, these cells do stick around and they do differentiate and do become part of the tissues where they are injected. So we have two completely different things here, right? On the one hand, we have allogeneic cells, which get taken out by the host killer T system, but they're there long enough to cause some tissue repair through paracrine signaling. On the other hand, we have autologous cells, which stick around, and I say stay and play, right? They don't get taken out because they're the patient's own cells. They do differentiate into other tissues, and these tracer studies show that very clearly. So hopefully this answers that question. I think it's a critical question for the field to understand. Thanks so much for watching. Have a great day.